Picture this. It's around 50,000 years ago, somewhere in ancient South Asia, and a woman is doing something that requires an almost supernatural level of calm given the circumstances. She has found a king cobra nest while foraging, which is the kind of discovery that produces one of two responses: immediate rapid departure in the opposite direction, or the very specific kind of focused stillness she is currently demonstrating. She has seen cobras before. Her group knows where they den in this territory, knows when they're most active, knows the specific posture that means a strike is imminent versus the defensive hood display that means the snake is scared and hoping you'll leave. This knowledge has been accumulated and transmitted across generations because in an ancient world where antivenom does not exist and hospitals are several tens of thousands of years away, knowing the difference between a scared cobra and a committed cobra is not a trivia question. It is a survival competency. What she's also noticed in the way that ancient humans noticed things as a matter of professional necessity is that the cobra that bit a smaller snake earlier in the morning and consumed it is absolutely fine. The venom that just killed a snake is doing nothing observable to the snake that produced it. And somewhere in the accumulated oral wisdom of her group is a version of the question this video exists to answer, because people who live in close proximity to venomous snakes for their entire lives across many generations inevitably notice that snakes seem immune to the thing that kills everything else, and they want to understand why. So here's the proper investigation. Why don't snakes die from their own venom? And what does understanding the surprisingly specific, surprisingly elegant answer tell us about how evolution solves the problem of being a biological weapons manufacturer that has to store its product safely, and why ancient humans were both correct and incorrect in some of their intuitive conclusions about how this works? Stick around, because this gets considerably more interesting than the simple version most people have heard, and the ancient human perspective on snake venom adds context that changes how you think about the relationship between predator, prey, and the chemical systems that mediate between them. Start with the most important clarification in this entire conversation, because the popular version of this topic usually gets it slightly wrong in a way that matters for understanding the actual biology. The common statement that snakes are immune to their own venom is accurate in the practical sense, but not quite precise in the mechanistic sense. Different species of venomous snakes have different levels and types of resistance to venom, and the mechanisms underlying that resistance vary considerably across snake families and venom types. Some snakes are resistant to their own specific venom through specialized physiological adaptations. Some are resistant to a broader range of venoms, including those of other species they encounter. Some are less resistant than popular understanding suggests. The relationship between snake and venom is more nuanced and more interesting than a simple blanket immunity claim captures. Begin with the delivery mechanism, because understanding why snakes don't poison themselves through normal venom production and storage requires understanding how venom actually gets into a target in the first place. Snake venom, despite what many people intuitively assume, is not toxic when it simply contacts tissue. It needs to be introduced into the bloodstream or lymphatic system to cause the systemic effects that make it dangerous. This is why humans can theoretically swallow small amounts of most snake venoms without significant effect, provided there are no cuts or lesions in the mouth or digestive tract through which the compounds could enter circulation. The venom glands where snakes produce and store venom are specialized structures derived evolutionarily from modified salivary glands, and they are sealed, specialized organs lined with tissue specifically adapted to safely contain venom compounds without those compounds being able to enter the snake's own bloodstream through the gland walls. The venom sits in the gland in the same way that any secretory gland stores its product, isolated from systemic circulation by the specific cellular architecture of the gland itself. A snake that never bites itself has no pathway for its own venom to enter its circulatory system, which means in normal circumstances the question of venom resistance doesn't actually arise through self-injection. The fangs that deliver venom are hollow or grooved channels that conduct venom from gland to target during a bite, and the tissue surrounding those fangs in the snake's own mouth is not a significant concern for self-envenomation, in the same way the gland tissue isn't, because the venom is in transit toward an external target during a strike rather than pooling in contact with vulnerable tissue. A snake biting itself, which does happen occasionally and is always worth watching if you happen to find it on video, can potentially introduce venom into its own circulation, and what happens in those cases is more interesting than the simple immunity story suggests. For the specific subset of snakes that have evolved genuine physiological resistance to their own venom compounds, the mechanisms involve several different biological strategies operating at different levels. The most direct mechanism is molecular modification of the specific receptor proteins that venom compounds target. Many neurotoxic venoms work by binding to specific receptor sites, particularly acetylcholine receptors at neuromuscular junctions, blocking normal nerve-muscle communication and causing paralysis. Some venomous snake species have evolved modified versions of these receptor proteins, where the specific binding sites that venom toxins attach to have been altered at the molecular level in ways that prevent the toxin from attaching effectively, rendering the toxin unable to produce its normal effect on the snake's own neuromuscular system. This molecular modification strategy is one of the more elegant evolutionary solutions in the entire biological world because it operates at the most fundamental level of the problem. Rather than trying to neutralize or break down the toxin after it enters the body, the modified receptor simply doesn't recognize it as something to bind to. The toxin that completely disrupts nerve-muscle communication in a mouse or human floats past the snake's acetylcholine receptors without engaging them, because those receptors have been modified to be specifically non-receptive to the toxin's binding sites while remaining functional for their normal biological purpose of transmitting nerve signals to muscles. The king cobra, which our ancient woman in the opening scenario was observing with the appropriate level of respectful attention, eats other snakes including other venomous species and has evolved resistance to a broad range of elapid venoms, the family that includes cobras, mambas, and various other neurotoxic species, through exactly this kind of receptor modification. Ophiophagus hanna, the king cobra's scientific name, translates roughly to snake-eating serpent, which is admirably literal, and the resistance to other snake venoms this hunting lifestyle requires has produced some of the most studied venom resistance mechanisms in snake biology. Mongoose species that hunt venomous snakes, including cobras, have independently evolved essentially identical receptor modifications through convergent evolution, arriving at the same molecular solution to the same biological problem through completely separate evolutionary pathways. This convergent evolution of identical molecular adaptations in two completely unrelated lineages, one reptilian and one mammalian, is one of the more striking examples in evolutionary biology of natural selection, repeatedly finding the same solution when faced with the same specific problem. Ancient humans who observed mongooses hunting cobras with apparently fearless disregard for the venom they were regularly being exposed to noticed this resistance without being able to understand its mechanism, incorporating it into oral traditions about mongooses in various cultures across the cobra's range. Enzymatic degradation provides another layer of venom resistance in some snake species. Venom compounds that make it into a snake's own bloodstream through a self-bite or through the experimental conditions that researchers use to study venom resistance may be broken down by specific enzymatic activity in the blood or tissues before they can reach effective concentrations at target sites. Some snake blood contains protease enzymes capable of digesting protein-based venom components, reducing their effective concentration faster than they can accumulate to dangerous levels. This mechanism is less reliable than receptor modification because it operates after venom entry rather than preventing the venom's effects at the receptor level, but it contributes to the overall resistance profile of many venomous species. Specialized serum proteins that bind to and neutralize specific venom components represent yet another resistance mechanism documented in various snake species. Some venomous snakes produce specific binding proteins in their blood that sequester venom compounds before they can reach target receptors, rendering them effectively inert. This is functionally similar to how antivenin works in medical treatment of snakebite, where antibodies that bind to and neutralize venom components are introduced artificially. In resistant snake species, the production of venom-neutralizing binding proteins is an endogenous capability that evolved through the same selection pressure that drove venom system development in the first place. The evolutionary logic of how these resistance mechanisms developed is worth examining because it addresses an interesting chicken and egg problem in evolutionary biology. For a snake to evolve resistance to its own venom, the venom has to be present first to create the selection pressure that favors resistance. But a snake capable of accidentally exposing itself to its own venom while having no resistance would presumably be selected against before resistance evolved. The resolution to this apparent paradox probably involves gradual coevolution of venom potency and resistance, with neither advancing dramatically faster than the other in the snake's own lineage, combined with the important point that normal venom storage and delivery doesn't actually require any resistance because the gland architecture prevents self-envenomation under normal circumstances. The most significant selection pressure for venom resistance in many snake species probably came not from self-envenomation, but from encountering the venoms of other snake species, either through predation of venomous prey or through encounters with venomous competitors. A snake that hunts other snakes faces immediate selection pressure for resistance to the prey's venom because unsuccessful resistance means death during prey consumption. This predator-prey dynamic provides a clearer evolutionary pathway for the development of robust venom resistance than the risk of self-envenomation alone, and it explains why the strongest venom resistance tends to be found in species with ophiophagus, snake-eating, dietary habits. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content, covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. Pit vipers, the large family that includes rattlesnakes, copperheads, and various other hemotoxic venomous species, have resistance profiles that reflect their specific venom chemistry. Pit viper venoms are primarily hemotoxic, disrupting blood clotting, damaging blood vessel walls, and destroying red blood cells through enzymatic activity, rather than producing the neurotoxic paralysis characteristic of elaphead venoms. The resistance mechanisms in pit vipers reflect these different venom targets, with pit viper blood clotting factors and vascular proteins being modified or supplemented in ways that reduce vulnerability to the specific enzymatic activities their venoms employ against prey. A rattlesnake exposed to its own venom shows significantly less hemotoxic damage than a mouse exposed to equivalent concentrations, reflecting this species-specific resistance. Rattlesnakes also provide one of the more interesting specific examples of resistance research because their well-studied natural history and the medical importance of rattlesnake envenomation have made them subjects of extensive investigation. Ground squirrels that coexist with rattlesnakes in California have evolved blood-borne proteins that neutralize rattlesnake venom specifically, allowing them to survive encounters that would be fatal for squirrels from non-rattlesnake territories. This prey-side venom resistance, evolving in response to predator venom, represents the same evolutionary dynamic playing out in the prey rather than the predator, and studying both sides of this chemical arms race has produced significant insights into how co-evolutionary dynamics drive molecular changes in multiple lineages simultaneously. Ancient humans navigating environments full of venomous snakes were effectively conducting empirical research on snake behavior and venom effects continuously across generations, building bodies of practical knowledge that were accurate at the behavioral and outcome level without access to the molecular mechanisms underlying what they observed. Understanding that certain snakes could eat venomous prey without apparent ill effect, that certain animals seemed resistant to snake venom while others were immediately vulnerable, and that snake venom behavior was specific and predictable rather than random, was practically significant information that ancient oral knowledge systems preserved and transmitted. The specific snake species that ancient humans in different regions had extended experience with shaped very different bodies of knowledge and very different cultural relationships with venomous snakes across different parts of the ancient world. Ancient human populations in sub-Saharan Africa navigated environments containing some of the world's most dangerous venomous snake species, including black mambas with a venom so rapidly neurotoxic that untreated bites are nearly universally fatal within hours, puff adders responsible for more snakebite fatalities across Africa than any other species, and various cobra species. The practical knowledge ancient African communities developed about these species, their habitats, their behaviors, and the specific circumstances that made encounters dangerous was sophisticated and specific in ways that directly affected survival outcomes. Ancient human populations in South and Southeast Asia dealing with the enormous diversity of venomous species in those regions, including king cobras, Russell's vipers, saw-scaled vipers, kraits, and many others, developed analogous bodies of practical knowledge calibrated to their specific local species. The specific behavioral signals that distinguish a cobra preparing to strike from one engaged in defensive display, knowledge that would read as surprisingly sophisticated animal behavior ethology if you described it in academic terms, was the kind of information that ancient communities in cobra territory maintained as practical survival knowledge rather than as academic curiosity. What ancient humans consistently noticed about snake venom immunity, and what fed into various cultural traditions and explanatory frameworks across different regions, was the observable fact that venomous snakes were apparently comfortable among their own kind in ways that contradicted the expected outcome of animals carrying such dangerous weaponry. This observation was accurate. The explanation that various ancient cultures developed for it was inevitably framed in the conceptual vocabulary available to them, rather than in terms of receptor modification and enzymatic neutralization, but the core observation was correct. Some ancient explanatory frameworks attributed snake venom immunity to a kind of internal fire, or special essence that snakes possessed, a magical or sacred quality ability that protected them from the power they carried. This is wrong as a mechanistic explanation, but represents an ancient attempt to conceptualize something real, specifically that snakes are not affected by their venom through some property intrinsic to being a snake rather than through luck or error. The intrinsic property explanation is correct in spirit even if wrong in specifics. Snakes are not affected by their venom because of specific molecular features of being a venomous snake, features that evolved as part of the same biological system that produced the venom itself. The specific case of venomous snakes eating venomous prey deserves extended attention because it represents the clearest observable demonstration of venom resistance that would have been available to ancient human observers. King cobras eating other cobras, which happens, provided direct evidence that the same venom capable of killing a human quickly did not produce the same effect when that cobra was consumed by another cobra. This observation is accurate because the king cobra's resistance to elipid venoms extends to venoms of conspecifics, other king cobras, and to the venoms of the other snake species it regularly preys upon. The implications of this for ancient human understanding of snake venom were significant. If venom could be swallowed without apparent effect by the snake that produced it, ancient humans who understood that swallowing small amounts of snake venom was less dangerous than being injected with it were operating on a mechanistically correct intuition, even without understanding that the difference between swallowing and injection was the presence or absence of absorption into circulation. Various ancient traditional medicine systems that experimented with consuming diluted snake venom were working in territory that, while extremely dangerous at any significant concentration, was not completely without mechanistic basis. The related question of why some mammals have evolved venom resistance is worth addressing because it provides important comparative context for understanding why snake venom immunity is not magically specific to snakes. We've already mentioned mongooses and ground squirrels. Hedgehogs show resistance to the venom of the European adder, which represents genuine adaptive resistance in an animal that regularly encounters adders in its foraging environment. Honey badgers, which are famous for their extraordinarily relaxed attitude toward being bitten by venomous snakes and then recovering, have been documented apparently recovering from cobra bites that should be lethal, though the mechanistic basis for their resistance is less thoroughly studied than some other species. Opossums provide one of the most interesting resistance cases because they have evolved a blood-borne protein that neutralizes a remarkably broad range of snake venoms rather than being specific to any particular snake family's venom, potentially reflecting a long evolutionary history of encountering diverse venomous snake species in the Americas. Research on this opossum venom-neutralizing protein has attracted significant interest in the context of developing more broadly effective antivenin treatments, since the challenge with current antivenin production is that it requires species-specific matching to the snake that caused the bite, while a broadly neutralizing compound of the type the opossum has evolved would be more generally applicable. The ancient human interest in understanding these resistant animals wasn't purely theoretical curiosity. In environments where snake venom was a genuine and significant mortality risk, any observable pattern about what protected some animals from it and what didn't was potentially medically useful information. Ancient traditional medicine systems across different regions developed various approaches to snakebite treatment that incorporated observations about venom-resistant animals, some of which had genuine pharmacological logic behind them even when the practitioners couldn't articulate that logic in modern biochemical terms, and some of which did not but reflected the human tendency to interpret observed resistance as transferable through some kind of physical or spiritual association with the resistant animal. The evolutionary co-evolutionary arms race between snake venom and prey resistance is one of the most studied examples in evolutionary biology of what happens when predator and prey are locked in a long-term selective cycle where each adaptation in one drives adaptation in the other. Snake venom has become increasingly potent across evolutionary time in many lineages, partly because prey species evolved resistance that made previous venom concentrations insufficient. Prey resistance increased in response to increasing venom potency. The result is a system where modern snake venoms are extraordinarily potent, not despite the resistance some prey species have evolved, but partly because of it, the ongoing arms race having driven both sides to more extreme positions than either would have reached independently. For ancient humans observing this system, without understanding its dynamics, the practical outcome was a world containing venomous snakes whose venom was highly effective against humans while being considerably less effective against some of the small mammals that shared the same environment. The differential vulnerability of different species to the same venom was observable and puzzling, and understanding why it existed was beyond the mechanistic tools available to ancient knowledge systems while remaining a consistent focus of curious observation across many ancient human communities communities living with venomous snakes as a daily environmental reality. The woman, in our opening scenario, watching the king cobra move through the undergrowth after its earlier meal without any sign of ill effect from the venom it had delivered, was observing something real and interesting that her accumulated group knowledge provided a framework for interpreting, even if that framework was conceptually very different from the molecular biology that actually explains what she was seeing. Her observation that the snake was fine, that this was consistently true rather than occasionally lucky, that certain other animals were similarly fine while humans were devastatingly not, was accurate at the level that mattered for practical survival decision-making in her specific environment. The full picture that emerges from examining why snakes don't die from their own venom is a story about co-evolution, molecular modification, enzymatic adaptation, and the elegant biological engineering that results when a lineage spends hundreds of millions of years as both the manufacturer and the potential victim of chemical weapons. The resistance mechanisms in venomous snakes are not accidents or lucky coincidences. They are the product of the same evolutionary pressures that produced the venom systems themselves, the two evolving in tandem as part of a single integrated biological system rather than as separate features that happened to coexist in the same animal. The ancient human fascination with this specific question, how does the snake survive what kills everything else, was a reasonable and intelligent response to an interesting observable phenomenon. The answers ancient communities developed were wrong in mechanism and right in observation, which is exactly the appropriate ratio of accuracy to expect from sophisticated empirical observation without access to molecular biology tools. The observation that something protects snakes from their own venom was correct. The nature of that something required another several tens of thousands of years of accumulated human knowledge before anyone could describe it precisely. Snakes don't die from their own venom because evolution had to solve that problem at the same time it was building the venom system, and evolution is very good at solving problems it gets enough generations to work on. The solution it arrived at, modified receptors, specialized gland architecture, enzymatic breakdown, binding proteins, is sophisticated enough to have survived hundreds of millions of years of snake evolution across thousands of species in environments on every continent except Antarctica. That durability is either the most straightforward evidence possible that the solution works, or is simply what adequate biological engineering looks like when you give natural selection enough time to refine it. Probably both. Definitely both.